Happy to introduce Lena Ting. Probably most of you know Lena. She's been a professor, full professor in biomedical engineering at Tech and Emory. Her lab is based at Emory now. Just a little bit of background about Lena. She did her BS in mechanical engineering from Berkeley, master's in biomechanical engineering at Stanford, and then also her PhD in mechanical engineering at Stanford. So California trained, and then went abroad, like some others here for her postdoc. Went over to Paris in the laboratory of physiology for a year, and then came back to Oregon Health Sciences University to do further postdoctoral training. She's currently the John and Jan Portman Professor of Biomedical Engineering at Emory. She's won numerous awards for research, for mentorship, for teaching, very extensive to list. She's actively involved in mentoring our students, junior faculty, highly engaging in terms of speaking, and we're very excited to have her here. And I'll just say one award that she won recently. She was elected as a fellow for the American Institute of Biological and Medical Engineering, which is a big deal. So Lena, take it away, multiscale interactions and muscle spindle.
Great. Thanks, Bilal. Hi, everyone. It's a pleasure to be able to come here and I'm talking to you about something a little bit different. I am not talking about balance control, although this work is very much related to that. It's a little bit weird to be given a talk right now given everything that's going on. So I just wanted to say I hope everyone has their power back. I hope everyone votes tomorrow. I've put here my Twitter handle, and I've found that over the pandemic and with everything going on, it's been a good way to connect to other people if you limit yourself to science. People to have conversations about science and about being a person in science. There's been some groups such as Black in Neuro and Queer in Neuro that have been become active and help people share their stories and hopefully feel a little bit more connected and included while we're all sort of socially distanced. So I'm going to talk about muscle spindles, right? And this is the sense that's not one of the five senses that allows you to touch your nose when your eyes are closed. It's one of the senses that is really taken for granted, and we, it's not one of the five major senses, and yet you don't know where your body is without it. Now, we've known about muscle spindles for a long time, since Sherrington and Ruffini, and they're these very, very beautiful, complex structures. And as we started thinking about muscle spindles, I thought, why don't people understand these? And I actually went back, and you'll see some pictures from the textbooks, and it's not necessarily because we don't know a lot about muscle spindles, but really, we don't necessarily understand what these signals are about. So here's some textbook signals. It's what we learn, right? Muscle spindles, they're these modified muscles inside your muscle. And when you stretch the muscle, they fire. And most of the work around muscle spindles is centered around this sort of stretch reflex, this tendon tap reflex, where you stretch a muscle, it fires, and then it has a monosynaptic connection back to the same muscle, causing it to contract. So that's the most fundamental sensory motor circuit that we can study. And then there's these other proprioceptors called Golgi tendon organs, which I won't talk about today. So this is what we learn. And I found that there's really a growing interest in understanding these signals because they don't just go back to the spinal cord, but they go everywhere. They go up to your brain. They go to other spinal segments. They go to your brainstem. They go to the thalamus. They help you perceive things. And I've been seeing a resurgence in people's interest in muscle spindles, especially in neural engineering, a lot of times from people who want to put on an exoskeleton and they want to know, what are the signals that we're generating? Are they realistic when we cause people to move? Can we simulate movements? We can do it biomechanically, but we don't have a model of the sensory motor feedback. And so we can't really make a reasonable model of what that transformation is between the sensory signal, because we don't know it, to the movement of the muscles. And so if we want to develop any kind of neural engineered solutions to either restoring sensation or generating movement, or using brain-machine interfaces to control robots and to send sensory signals back to people who have lost sensation, we really need to understand how we perceive our own bodies. So I've been having a lot of conversations with others that go something like this. Oh, you study spindles. What they want is a readout of muscle spindle signals during motor behavior. Right? So it's like, hey, Lena, is it okay to model my muscle spindle signals based on muscle tendon length and velocity? Because I've got this video-based motion capture, or I've got a simulation, and I want to move my robotic device. And I say, well, that's probably not going to work that well, especially during movement. And I get, well, what about as a first approximation? And then I'll say, well, no, I don't think so. And they come back with, okay, well, what else do we need to know? Like, how complicated could it be? And so then I talk about the complexities of muscles, spindles, and gamma drive, and it probably turns into like a, wah, 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 wah thing, which it was for me too. And it's even worse because most of the things we need to know, we have never been measured really robustly during movements. And so I get something like, okay, I'm just going to use life. So I've seen this in lots of different papers recently. And so, really, we still really lack a fundamental understanding of muscle spindle function in naturalistic movements. So we went to start studying the stretch reflex, and just to simulate when you hit the tendon, how far does your knee kick out? And it turns out there are no successful simulations of this really simple knee-jerk response from a neuromechanical perspective, which basically means we don't know what the signals are generated during that most fundamental circuit. So how sensory motor behavior, so how could we really think about modeling or understanding more complex movements that, require intention and things like that. So, part of the problem is our accessibility to muscle spindle afferent signals, as well as these gamma motor neuron efferent signals to the spindle itself are still inaccessible. There's no, there's no equivalent of an auditory nerve for muscle spindle proprioceptors. Generally, if people record them, they don't really know what kind of sensory signals are recording. And, I think hopefully that with the advent of optogenetics and other types of ways to manipulate the sensory and motor system, that we'll be able to gain more access. But right now, those things haven't yet been developed enough for us to understand those. So I think we're still a ways from understanding that. And even further, that impaired muscle spindle function is implicated in a number of movement disorders, from sensory neuropathy, which is just primary damage to those afferents, to chemotherapy-induced peripheral neuropathy, which might maybe change just the neuron, aging-related soft tissue changes the sort of mechanical environment, the extracellular matrix, the tendon, which also changes muscle spindle signals. Muscular dystrophy and other things can change the kind of muscle myosins that are expressed within these sensors. And then there's a whole class of symptoms of neurological motor disorders now falling under the category of hyperresistance, joint hyperresistance. This includes spasticity, which is manifest in stroke, spinal cord injury, cerebral palsy, and many other types of diseases where people get stiff limbs, and really don't know much about these, except that these are neural in origin, and many of these are things that people are trying to correct with drugs or robotic devices, surgery. And those are all just sort of primary types of things. The spindles are involved in sensory integration, higher level processing, motor planning, and perceptual deficits. And so all these changes are going to go upstream. All right, so what I'm going to talk to you about today is really, first, we have many hundreds or so years of study of muscle spindle firing, but these are mostly descriptions. And so they're very specific to the particular conditions under which data were recorded. And then I'll talk to you about key mechanisms. Can we gain a better mechanistic understanding of these? And we've looked at, okay, the muscle fiber inside of the muscle spindle, integrating both, central commands and peripheral interactions. Whoops, that one didn't work. And then to say, well, how do we build, excuse me, the simplest predictive model to understand these kinds of naturalistic behaviors where we can't really record it right now, but we could maybe generate some predictions about what should happen, and then really kind of going meta of like, okay, we really understand what these signals actually mean during movement. So I want to just note the people who actually did this work. It's lots of multidisciplinary collaboration with very difficult conversations because we all have different training and vocabulary. So I started by going back to the lab where I did my postdoc in Paris to start this project after just not being able to understand the motor behaviors that we study. Kyle Bloom did a PhD, and most of the work here will be his work. He's now studying spindles in primates with Lee Miller at Northwestern. We had to pull in a muscle biophysicist, spinal cord electrophysiologist, Tim Cope at Georgia Tech, and Brian Horseland, a postdoc, worked on this project and is now starting his own lab. So back to the spindle. Here's the figure that many of you have to draw for your qualifying exam. So a spinal cord with an afferent, and then causes a muscle to contract, right? Let me get some water, excuse me. Okay, so hopefully you've stared at that long enough to pass your qualifying exam. The part that's not on your qualifying exam is the fact that the muscle spindle itself is an extremely complex organ. And it's made of muscle fibers. So it's a sensory motor organ. It's not just a sensory organ. So here's a bunch of fibers inside of there. They're called bags and chains. And there's muscle fibers on both ends. And then there are motor neurons going to those muscle fibers. And so we call that gamma motor neuron activity. There's gamma statics and gamma dynamics, and they project to different parts of the spindle. And then the signals that we get are actually due to these 1A afferent. So the sensory neurons are wrapped around the middle of these modified muscle fibers. Right, and there's group 2 endings, which I'm not even going to talk about. All right, so it could be that like this motor activity going to muscle spindles is even more complex than the motor neuron activity going to the muscles that cause the movement. So we have a muscle within a muscle, and it's really well centered for integrating the sort of central and peripheral motor control mechanism. So I took this out of the textbook. It's in the textbook, but you forgot it. Because it's basically a bunch of facts about muscle spindles that are really hard to sort of memorize because we don't have a great understanding. So you're going to have alpha motor neuron drive to an extensor muscle to a flexor muscle, and then that's going to, the force and the movement are going to depend on the inertia and all kinds of biomechanical properties of the muscle. So that's going to affect what the muscle can sense. And then within one of these muscles are a whole bunch of muscle spindles, and they're distributed throughout the muscle might be a couple hundred, and then they each get their own motor command. And then here's the more complex picture that I showed you, where the commands go to the static and the dynamic. And so overall, if I were to write like, okay, what's the firing rate, instantaneous firing rate here of a muscle spindle related to, it's basically related to all kinds of length, velocity, acceleration, force, and stiffness of the muscle, the alpha drive, the gamma drive. So it's a complex function. And case in point is that you can create an experiment to have the muscle spindle look like any of these variables, right? So so here's a pretty classic. This is also from the textbook, so it's there. So when you just, here's a muscle, you stretch it, and at the top here is sort of what you get, right, for firing rates, impulses per second. And this is why people say it has something to do with length, is because once you stretch it to this new length, you have a higher firing rate. It's also related to velocity, because during the stretch, so during this ramp, you also have sort of an elevation here that looks like there's summation of length and velocity. And but then if you stimulate the gamma static motor neuron, then what you do is you elevate the baseline firing. If you activate the dynamic gamma, so motor neuron, then you change sort of this velocity sensitivity, but then this is complicated here. It's not related to either. So these are very controlled experimental conditions, but they're experiments in humans where you can impale a muscle spindle and you can measure it. The recording as you passively move the ankle around or something like that, and in this really neat paper by Valerie Hospod and Edith Ribot-Siskar, they showed that you can change the sensory signal coming out of the muscle spindle by asking people to pay attention to different aspects of the movement. So if you say, how fast are you going versus how far are you going, you actually change the signal coming back. And then this is in the textbook too. So when you're actually shortening your muscle, so you're sending a command to the alpha motor neuron and the gamma motor neuron, and they get integrated based on the load, the external load, and the length. But here's an example from a human again of a muscle being shortened and the muscle spindle being activated. So this is impossible if you don't have gamma drive, because you're shortening the muscle and the spindle will go slack and it shouldn't actually fire. So one of the things is that gamma motor drive may reflect sort of motor intention and sort of help you, depending on the ratio of alpha gamma, I'll get into it later, you could have firing during shortening and lengthening. Okay, so lots of times people say, well, if I just know the muscle is shortening or lengthening, can't I just model the muscle spindle sensory signal? And so case in point here, in this case, you would get absolutely the wrong thing. Okay, so during locomotion, natural movements, we don't have very many things, because basically these human experiments, you can't move very much because they have to like stick a needle in your arm and only very small and slow movements. But there have been a few recordings in, this is a cat, where, okay, here's the muscle activity, which is a proxy for sort of alpha motor neuron. Motor command that causes the movement. Here's the length of the muscle based on joint angles. And then here's a recording of a muscle spindle 1A afferent, so that's the one we're going to talk about the most. And you can see that it doesn't really look like length. Maybe it could be velocity, but then when it stops moving, we still get firing. In some of their models, they have to use the alpha drive to model this activity, but here you can see that even when the EMG is on, you get some activity in this phase and you don't get activity there. So it's very complex. And then there's been, so here's another case during locomotion. This is in a decerebrate cat where, again, we have two muscles firing. And here's the ankle. angle. And here you see these impulses, and oh wow, it follows the ankle angle really well, right? So that would be a case for this sort of readout. But this is actually the gamma drive, the gamma static drive. This is not the afferent signal. This is the command going to the spindle itself. So hopefully I've convinced you not to use just length to model your muscle spindle. And to me, this means that there's some internal model going to the muscle spindle motor command that has something to do with what I expect how I expect to interact with the environment. All right, so what are the key mechanisms underlying these relationships? So far, I've just listed a bunch of facts. It's sort of like, stamp collecting. And now I want to understand how could I account for all of these really crazy relationships in a mechanistic sense. And so one of the data that I got in France was from really classic experiments again, anesthetized animals, passive, so there's no motor drive to the muscle or the spindle. And so that experiment's been done thousands of times. But what we were looking in particular at was not just the length of the muscle, but also the force of the muscle here, and then the yank. What we're calling yank is the first time derivative of the force. And what we noticed here is that the sort of temporal signature of the yank looks a lot like the firing rate of the spindle that we recorded. So you can see this burst and a ramp, and then this sort of decline here looked a lot like this decline in the force. Okay, so and it was suggested in the 70s that maybe muscle properties like this were actually driving muscle spindle firing, but that sort of got dropped because it just wasn't technically feasible, and it was never recorded at the same time and recorded. And so the idea was there, just not the technology. And so we did something really, really simple, and we took the recorded force and the yank from the muscle, we added them together, rectified them like they're passing through a neuron, and then looked at whether we could predict or reproduce the muscle spindle firing patterns that we recorded, and then we used sort of acceleration, velocity, and length, which is more classic, although acceleration has been debated, to reproduce that. And the most striking result here is that if you stretch a muscle several times, the first time you stretch it, you get more firing than the next time. And you really can't account for that using the kinematics itself, but if we measure the force and the yank, we see that really it's just the force of stretching the muscle is bigger the first time. And that force actually just, and the yank really just accounts for the difference in firing the first time versus the next time. Okay, so other times it looks pretty good. And if you try and reproduce some of the classic experiments and you look at the bottom here, the kinematics looks really good. So this is a length and velocity in red here, really predicts the firing rate pretty well. I would totally report this in a paper. And then as you go faster, it starts to fall apart here. But with the same parameters of our model, we don't have to change anything. We can account for these more complex changes in muscle spindle firing if we have the force and the yank of the whole muscle, which isn't really quite right, as I'll show you. But it was a proxy for like all the spindle and the muscle are all doing. So that's our assumption there. And then Tim Cope and Richard Nichols had written a paper really outlining that this weird property, this history dependence of the muscle spindle actually was related to fundamental kind of biophysical properties of the muscle. So Tim had this experiment where you stretch the muscle 3 times the same way, and the first time the muscle spindle gives you a burst and a sort of higher firing rate with a different slope, and then the second time it changes, and so it's different. So again, there's no one-to-one correspondence between the muscle length and the firing rate, and this behavior is dependent on the muscle sitting there quietly for a while. And around the same time, Ken Campbell, now our collaborator, also did this experiment that in a isolated skinned single muscle fiber that's put in a calcium bath, and he stretched it 3 times with a little bit of a break, and he varied this, and he showed that the muscle fiber itself had a hitch in the force the first time, and then it went away, and the second time you stretch it, it's different. Okay, so these are extremely parallel. They never met till I introduced them, and then but these are very, very interesting. And Ken is a biophysicist, a muscle biophysicist, and so he was able to develop a model based primarily on actin-myosin cross-bridge interaction. So you probably learned that in physiology class with the myosin head attaching and detaching to the actin, and it's definitely not a level of muscle modeling that I was particularly interested in pursuing until I saw this. So with that, we said let's build a model to do all these things. All right. So to do that, then we basically, whoops, wrong direction. There we go. Let's build a biophysical model of this muscle spindle based on the fact that it's a muscle. That's a muscle that has actin and myosin, and there's 2 types. Well, I showed you 5 fibers, but basically we're going to do 2 prototypical muscle fibers. One's a bag fiber and a chain fiber, and they have very different myosin types, and this really changes the kind of firing they get. But the bag fiber is the one that gives me really dynamic spikes, sort of bursts of activity that we're really interested in. Okay, so you get these rate equations for the 2 spindles, and we take the force and the yank of one of the fibers, the force of the chain fiber, and we add them together basically to generate like a driving potential for the muscle spindle afferent neuron. Okay. So during a passive muscle stretch, everything's just sort of kind of attaching and detaching very, very slowly, and we stretch the muscle spindle, and we assume that the muscle and the muscle spindle are basically experiencing the same stretch, and so they'll have the same response. And what we were able to do is sort of vary. These are flipped now. Okay. So here's an illustration of sort of the driving potential to the neuron of the bag fiber. Looks like this blue thing, and then from the chain fiber it looks like the red thing, and then we add them together and you get sort of the driving potential, and if it crosses a threshold then it'll fire. And so depending on how we weighted these components, we could generate a whole grid of kind of potential muscle spindle firing responses to this classic stretch. So if there was a lot of dependence on the yank here, then we would mainly get these big bursts at the beginning of stretch. And the more the driving potential depended on the force, the more we were getting this sort of tonic firing at the plateaus. Okay, so as I go down, I get more tonic firing. And when you do experiments on muscle spindles, you can kind of classify muscle spindles with these different types of characteristics. So you might call this one a dynamic spindle, and this is a more static spindle down here. But we think that this whole family of sort of continuum of possible responses could be generated with these 2 sort of mechanisms. All right, so then what we did is we tuned the properties of the myosins in the model so that it would be this history-dependent response. So basically, again, we're going to stretch the muscle twice and then wait for 10 seconds or wait for shorter periods of time. And as you can see, if I stretch it once, I get this sort of response. The second time, I get a very different response. If I wait 10 seconds, it comes back. And the less time I wait, there's a different amount of firing. So, and this, because we have a model, we know this corresponds very directly to sort of how many actin and myosin molecules there are that are connected, how likely they are to be to be attached or detached. So it really just comes out of these rate equations for the attachment and detachment of muscle, actin, and myosin. And so we basically, our model, it recovers after a period of time, and there's similar but not quite exact recovery in muscle spindle firing that also follow this characteristics that's, again, been known for a very long time. Okay, so then we said, well, let's just try all the experiments that have been done. So here, what's emergent from this is that if you increase the velocity of the stretch, then you get these different profiles of firing rates or driving potentials. And people generally classify these as the initial burst, which is the height of this, and then the dynamic index, which is like what this firing rate here is versus half a second later. Is somewhat. One of the things that came out is that the first burst looks like it scales linearly with acceleration, which is something that has been shown before, although somewhat contested, and also really relevant to the sort of responses to perturbation that we see in balance control. So we see the reflex or the muscle response is also linearly related to acceleration. The second is that this dynamic index, again, it's this ratio of the burst to this response, increases non-linearly with the velocity of the stretch. This has also been known since the 60s, and this has been fit as a fractional power law. And in other models, people have explicitly put in this sort of nonlinear relationship to model muscle spindle output, but we didn't do that. It just really emerges from the dynamics, again, of the actin and myosin. So we didn't have to tell it to do this. It just sort of emerges. And so that gives us some confidence in our model that we actually have a mechanism that generates lots of things that we didn't set out to explain. All right, so again, now if here's our real complex model, and you can activate the motor neuron going to the dynamic bag fiber or static one. And again, these are heroic experiments done in the 60s. They'll show that, okay, with no gamma drive, and if you stretch the muscle at different velocities, you can get these different firing patterns, right? If you then stimulate the gamma static, okay, gamma motor neuron, you change the muscle spindle firing pattern upon the same stretch. And if you activate the dynamic one, you change it in a very different way. So again, without knowing what the gamma drive is, it's really impossible for us to predict what the muscle spindle is actually doing when you observe a particular length change in the muscle. So we modeled that by changing the probability of the myosin attaching to actin in the bag versus the chain. So the bag fiber dynamic and chain fiber more like gamma static. And so what we get is, so as we increase bag fiber activation, just like in the experiments, we tend to change the response during the stretch and not so much during sort of hold periods. If you activate the chain fiber, so that's gamma static, you tend not to change the response to stretch, but you change sort of the tonic background level of firing at a particular length. And then I'm not going to go into it, but there's different like properties of scaling and those types of things also, as we increase stimulation, the scaling properties that we saw happen to look like what was shown in the 60s. Again, giving us some confidence that we have the mechanism right of what this potential is. Okay. So, all right, that's fine. Those are still really unnatural experiments. Here again, we go back to the textbook and it shows this muscle spindle in parallel with the muscle itself. And then again, if the muscle contracts and the spindle goes slack, and then it stops firing. And then if you activate it, then you can get firing there. And so we needed to simulate this sort of higher level mechanical interaction, which we did here, where the muscle spindles, you get alpha motor neuron drive to the muscle, you get gamma motor neuron drive to our model of our two different kinds of muscle spindle fibers, and we attach it to a tendon. So that allows us to kind of simulate this interaction now. All right, so here what we're going to do is we're going to keep the end-to-end length here the same. So when you contract the muscle, the tendon's going to stretch, the spindle is going to go be the same length as the fiber, and then what we're going to do is first activate just the extrafusal fiber, then just the intrafusal fiber, gamma, and then both in different proportions. So there's a little simulation where you can see that as you activate the all right, so what do I have here? Here's the amount of activation to the gamma, first one, and then here are the change in length of basically the end-to-end length doesn't change at all throughout the whole. And then here's the firing that we get of our motor neuron. We just pass the potential through an integrate and fire. And then this is a stress or the force in either the intrafusal or the extrafusal muscle. So here you can see that when you just activate alpha, you're going to silence the spindle. When you just activate gamma, you can drive the signal. And then depending on the ratio of alpha and gamma, you can either have it be silenced or activated. And again, this is pretty much the same length change. The only one of these variables that, by design, is always corresponds to the firing is the force in the intrafusal fiber. So it's not length, and it's not extrafusal. So that then might explain. Oh, this was supposed to go. Some of the weird things we see when we start having humans generate force. So here, if a human just like pushes with their finger and generates a torque, and there's no end-to-end length change of the muscle, you can get a condition here where basically you get depressed firing of the muscle spindle, or you can get a case here, the torque looks the same, where you get increased muscle spindle firing, right? And so we can simulate those in our model. Here, if you don't turn on gamma at all and you increase the amount of contraction or the amount of, basically just reduce the firing. Then depending on if I just activate gamma in the same ramp, it really depends on the ratio of the two. And sometimes I can get increased firing, sometimes I can get no change in firing, and sometimes just change in firing. So again, this is why it's really important for us to measure the gamma drive to the muscle spindle in order to understand what is the signal what's the sensory signal arising from it. Okay, and then if you stretch the whole thing together, so here we stretch them with those classic ramp and holds, depending on alpha drive being off or on, gamma drive being off or on, we're going to see really different firing rates. And in particular, this burst, I think, is really important for detecting contact events. So if you're going to pick up an object or somebody's going to hit you, then you really have to have the spindle be active, or you're not going to get very good signals related to those things. Okay, so we have a model, and it's got some hierarchy there. We don't have all of the details in there, but I think it's a framework where we can start to add different things that you might have questions about, like how stiff is the muscle or the tendon, or what kind of ion channels are there in the encoding region, what happens when the extracellular matrix gets stiffer. So we think that it gives us an ability to start to interface with some of these actual physiological impairments that happen with chemotherapy, with various genetic diseases and aging, right? So, but that still doesn't tell us, like, what the hell are muscle spindles encoding? Like, what does it mean? Why is it changing all the time? And so one of the questions is a muscle spindle like an internal model, a physical internal model, muscle and body environmental interactions? And so here I've got, this one of my favorite versions of the well-pert, why can't you tickle yourself thing is when when you send a motor command, you also send efferent's copy to a predictor, and here it would be a neural predictor. And if there's a discrepancy, right, then I laugh because it's ticklish. So if I didn't send this signal and somebody else tickles me, then I'm ticklish. But if I know that I'm tickling myself, then I get no signal here at all, right? And so it's the same kind of idea there, except that, maybe that predictor is actually happening not in a neural substrate, but in this muscle, this modified muscle, right? And then the results, the impact here is not just, I feel ticklish or not. It really is very critical to survival. Like, you're going to fall down, or you're going to get eaten by an alligator or something like that. And this also explains why muscle spindles have been shown to play a role in perception of, how heavy an object is, a muscle fatigue, force, and position. And these are things that people are like, well, is that really true? And it's sort of still unexplained. And all of these conclusions were drawn very indirectly from behavioral experiments. But I also want to say, in spindles, we have more than just efferent's copy. It's not just a copy of what you sent to your muscle. So so it turns out that, okay, you have alpha motor neurons, which go just to the muscle. You have gamma motor neurons, that just go to the spindle. But you actually have a whole class of beta motor neurons that go to both. So that's where your efferent's copy is, right? You get the exact same signal going to the spindle and the muscle. And it turns out that probably, it's estimated that about 30% of neurons sort of identified electrophysiologically as alpha are actually beta motor neurons. And so the fact that we have gamma and other animals like amphibians and fish don't suggests that gammas really have some other role beyond just efferent's copy. Like, there's no way we would have developed these things if they were just going to do the exact same thing as beta. Okay. So is a muscle spindle an internal model of a muscle, and maybe it's an external, it's a physical model? There is some homology to, not homology, that's the wrong word. It's sort of a parallel to what happens in houseflies. So a lot of insects have two sets of wings. Usually both are used for flying. But in flies, the halteres are these degenerate little tiny wings, and they don't contribute to flying at all, but they're sensors. Okay, so in the same way, we could think of the muscle spindle as a sort of degenerated muscle, but that then really there to provide information about the environment. And so these are parallel because they both receive motor commands, right? So these little wings flap while the big wings are flapping. And then, and if there's some sensory signal, like a gust of wind or something, that actually directly changes the motor output, just like the stretch reflex, right, that we've seen, plus higher level things. And then the sensitivity then is modulated by descending commands. So if you flap in a slightly different way, or if you activate your muscle spindle in a slightly different way, then you're going to get a different signal. Okay, so I think the muscle spindles are really primed to encode these interactions between the body and the environment, and in particular ones that are really hard for us to compute. Right, so if you're balancing, or you're picking up an object, or you're in rough terrain or in water, this is really, really tough. So So, based on what you want to do, you have to develop some kind of internal model or expectation about how your interactions with the world are going to happen, and that's going to be sent to the muscle to generate the movement, to beta alpha motor neurons, to beta motor neurons, but also to gamma motor neurons that are going to adjust this sensitivity of the sensory organ. And those gammas are also going to be affected by attention or state, right? Am I in a heightened state of awareness? Am I going to fall off a cliff? Am I wearing shoes? So all of those things are going to affect, what the spindle signal actually is. And so Peter Ellaway, they have said, oh, the static gamma seems like this temporal template of intended movement. So it actually seems to kind of follow just the movement pattern that we expect. And it seems like the dynamic gamma, which gives us those bursts, might detect the onset of muscle lengthening. Right? So that's where I think those contact events, when my foot hits the ground, is right. And they say, oh, and it detects departures from the intended movement trajectory, which I think really is the role of the static. And one thing I want to say here is that, thinking about movement as just movement trajectory is not, is just one aspect of movement. And this interaction and exchange of power and energy between the environment and the limb and sort of just touching things is also extremely important. Important to understand. So, like, if I want to be precise about picking up the cup or landing on a rock, it's not just about my trajectory, right? And the thing that with the flies, well, I'll just do this with so as the body of animals gets and the environment gets more and more unstable, we start to see more complex muscle spindles evolve. So fish have one intrafusal fibers. Amphibians only have beta, they don't have gamma. And then I think as we go to more and more upright animals in this gravity environment, we start to see more complexity of the control of muscle spindles. And, these environments that we're trying to understand are ones that we actually still can't simulate. So ground contact events are really hard. If you're a fly, the aerodynamic of flies flying around is something that as engineers we have a hard time simulating. So why actually think we could generate a simulation in our brains? You would actually just have this little physical model, and that's actually how a lot of robotics is done now, because we can't model the events. You actually have to do a physical implementation to understand what's really going to happen. And case in point here, people without proprioception can't account for different biomechanical features of their arm during reaching, so they don't really know what the anisotropy of the arm, and so they start moving in a way that is not taking into account those interactions of physical forces. So just to wrap up here, I want to say that the muscle spindle is not just an afferent signal; it's a sensory motor signal that integrates a lot of different aspects of movement. And so maybe we shouldn't ask what do muscle spindles encode, but more what do they compute, right? And so the central effects on muscle spindles could include attention to different features, anticipation of events, what our intention is, the strategies we have based on expectation, like what are we going to encounter in the world, and in that sense, I think that the signals of the gamma motor neuron are probably learned and may differ according to an individual's experience. Those are my hypotheses that yet remain to be tested. That's the same slide. And so I just want to say, like, we really need people to work on sort of more of a mechanistic and theoretical understanding of what muscle spindles kind of do in order to treat and restore movement. And with that, I just wanted to thank you for your attention, the members of my collaborators.
Okay, there was no questions in the chat, but feel free to unmute and ask any questions.
Hey, I had a question. Overall, that was a really interesting and great presentation, Lena. Thanks so much for laying that out. Um, I was wondering, given kind of the role you see muscle spindles playing in terms of, active movements and interaction with the environment and that sort of thing, do you think there should be, or is there, this is, I'm not familiar with this literature, is there a big difference between, what muscle spindles should be doing at the digits, where we obviously interact with objects, versus what they should be doing in the limbs?
Yeah, so that's a good question because, and we have to think about what are the physics of the hand versus the limb. And one of the things, and I talked to Sydney Chestek about this when she came, is that a lot of the control of the hand is different than of larger limbs because, if I flex my hands, they flex whether my hand is like this or like this. And that basically means that the inertial part is not so great. So F equals ma is not so important there. Whereas if I'm swinging my arms and I raise my hand like this and I turn over, it's a completely different muscle. So that actually simplifies things a little bit. So now you're dominated by viscosity. Force equal is more proportional to velocity in this case. Rather than acceleration. So it's one less integration, which means that it's, first order differential equation rather than a second order differential equation. And so its dynamics are a little bit more straightforward, but I think then contacting objects. So we saw Adam Hampman, like mouse grabbing pasta is not a trajectory task, right? It's all about this grabbing the pasta, and then the other is that there's the stiffness in the hands, right? So it could be that you want to maintain a certain bias force on the pasta, right? So it doesn't slip, right? So it's really not about how much I'm moving, but how, am I applying enough force to keep this thing in my hand? So even though what we remember from class is that it's length and velocity. There's lots of examples where muscle spindles appear to follow force, muscle activity, basically every variable that you can think of. And, I have that function, and you've basically crossed off different aspects of that function under those conditions.
Lena, that was really cool. I'm glad to hear the sort of full elaborated story because I think I've heard bits and pieces of it coming through.
Well, I'm glad I finally made it. So.
Yeah. The So a lot of the stimuli that are still used and are still sort of quasi-static in some regime. They're still sort of like you're still keeping velocity constant or having these gaps in between. And you had a lot of really brilliant examples of sort of introducing different time history and that kind of thing. But even in the fly literature, for example, a lot more people are doing now sort of more dynamic characterization, which of course when it's nonlinear has its own drawbacks. But I'm just curious if anyone's done more like frequency response characterization, white noise analysis, any of those kinds of strategies with this.
So, people did frequency response. We have a paper from the 60s where they did this, but it's in the absence of naturalistic alpha and gamma drive. And so, because it's all done in these anesthetized preps, or where they had to, because there's a mixed nerve, they literally had to dissect out micro bundles of the axons, the motor axons, and like hope that they got a gamma. And we still struggle. We don't have good ways of activating gamma even in an anesthetized preparation, and we don't have good ways of activating alpha in an anesthetized preparation because you stimulate. So we're still waiting for people smarter than me to like develop technologies to activate muscles and motor neurons in a naturalistic fashion. So more than on and off, right? Especially with gamma. So some of the optogenetics may be more binary, less, they need to be graded. We need to be able to keep track of them. And so, this is sort of I'm really happy to see motor control finally kind of coming back with all of these new techniques, and want to push further to say, like, even if we were to just record the firing of the spindles, we still wouldn't understand what was actually going on. So not just turning on gamma, but also recording gamma activity. Did you ask a question and did I answer it?
Yeah, I mean, you basically said that they had done it in the past, but without the context of the alpha and gamma
Yeah.
activation, it doesn't make a lot of sense. Yeah, I was sort of more curious in these new ways where you're doing it, if anybody had applied that, but I think you're saying that even technologically that's quite difficult to do. And it's a
Yeah,
little
right.
bit
Yeah.
to interpret, but you've got this nice model now that might make some nice predictions in you had some sort of quasi-static cases where you could show that it was the gamma force, the intrafusal force that was really what was reflecting the firing rate. I would be curious if that sort of extended to a more dynamic regime, especially given your argument that it's as the environment gets more complex, you get the drive now to have beta and you have beta and alpha.
Yeah, right. Yeah. So that would be really great. And, you think about if you're a fish and you mess up, you're not going to fall over. Once you get on land, then it's really important to have more nuanced kind of information.
Hey, Lena, Richard Nichols here. Hey, that was a really good talk. I enjoyed it very much, and you've done a prodigious amount of work. I remember Kyle as a rotation student in my laboratory talking about these issues all those years ago. I just have two comments. One is I completely endorse your idea that spindle is a physical model of at least of the muscle, if not more. I just wanted to remark that, in fact, this idea that the spindle is a predictive mechanism or a model reference system that idea was begun in 1981, published in 1981 by Jim Hauk and Zev Reimer and Pat Crago, and further elaborated by Jim in the late 80s. And I wanted to say that because Jim passed away this year, and
Thank you.
his work is really critical here. The second thing is that I just have one slight technical bone to pick with you about the use of force and yank. I would submit that force and yank are proxies for intrafusal muscle fiber stiffness. In other words, the length variable is still in there. And the thing is that I didn't see anywhere in your talk where you accounted for fascicular length changes. Everything was concerned about muscle tendon unit length changes. So I would say that if you see a change in force and yank measured at the tendon, it almost has to be the case that there's some excuse me, if there's some history dependence there almost has to be some history dependence in length changes of the fascicles. We might want to discuss this offline, but that's.
Oh, yeah. Actually, no. Our model does look at the fiber length, not the muscle tendon length.
Well, I know, but your measurement, I'm talking about your experimental measurement.
Oh, yeah, absolutely. I mean, we aren't able to measure the things that we measure.
Yeah, and then when if you were actually able to manipulate the length changes of a single muscle spindle, then the spring that you'd be looking at is the membrane where the receptor is, right? And so again, I think in other words, this is kind of technical, so we don't need to go.
Yeah, it's I'll just say this: we didn't have enough information. The central part of the intrafusal fiber. We assumed that it was a linear spring, but it's a bag, it has viscosity, and we also don't know much about the ion channels in the neural fiber the afferent wrapping around that. So there's a whole, so people have asked us to model it, and we just said we really can't find enough information to do it. So all we know is that the force and the yank of the intrafusal fiber has a very, very close relationship to the firing rate that we measure. And there's a whole bunch of stuff in between that that we don't yet know, and that we need people to study. And I think people really wanted to get into the biology of it. There's tons there, but like ion channels haven't been identified. I'm sure there's like integrins hanging on to, with the neuron onto the fiber, and then probably a much more complex dynamics of the encoding region itself that again, like now we have to do micromechanical experiments on these things in an intact animal. So I've talked to some of the people at IBB about these like molecular measures of forces and things like that. But these are all in vitro preps. And so again, like we wouldn't be able to know what was happening during that movement.
Great, thanks.
Okay,
As
I'm
far
going
as
to
the
jump in there. Sorry, I'm going to jump in there because we're already 3 minutes over our time. I'm glad there's lots of enthusiasm about the talk, but please take advantage of the fact that Lena's a local speaker and reach out with further questions. And thanks again to Lena for giving a talk that's got
Thanks.
people fired up apparently here at the end. All right.
All right. Thank you.
Thanks, Lena.